All right. So session number 2 is going to focus on speakers from academia, industry, and nonprofit organizations. To start us off, Dr. Tim Yu will start. Tim is a physician scientist at Boston's Children's Hospital, and he's going to talk to us today about gene-targeted therapies for the long tail or orphan diseases. Okay, Tim, are you with us?
I am, Wayne. Thank you so much for having me. Can you hear me?
Yes, we can.
Okay. Yunwei and Wayne, I would like to thank you for hosting this event in honor of Rare Disease Day and for bringing some of my favorite people together. Hello to many familiar faces, and glad to have the chance to spend this time with you. I'm going to provide a brief overview of what I think are really exciting opportunities in the rare disease space. This is an area that's seen tremendous change over the last 5 years, and all of you are a part of it. This will hopefully be a helpful overview, although I suspect that that much of the details for what I'm going to share are at least somewhat familiar to many of you in the audience, but I hope that it will provide a framework for some of the questions and discussion that might ensue. So my talk is gene-targeted therapies for the long tail of orphan disease. And just by way of background, where this is coming from, I'm a neurologist and geneticist at Boston Children's Hospital. I have the privilege of working with a really wonderful, dedicated team of some 20 people, researchers, clinicians, MDs, MD PhDs, and PhDs, and research assistants in our group who've been dedicated to this really new space of genomic medicine for the last 5 years. And the question that drives us, I always show this slide, is that many of you who've joined this meeting recognize that the diagnostic odyssey is just the beginning. And once a diagnosis is achieved, we are faced with the fact that now the question is, now what? And we all recognize that despite how incredibly important it is to now know the names of our children's conditions, that treatments for these conditions currently remain too scarce. Luckily, we're all motivated to actually span this gap. The fact that there are only 500 current rare genetic diseases that for which there exists some sort of therapy. We're motivated to span this gap, and that there is progress being made. There's a growing wave of genetically targeted therapies, gene therapies, mRNA therapies, antisense oligonucleotides. Someday down the road will be coming CRISPR editing and other siRNA-based technologies, et cetera. The question is, when will it come for us? We have been working, as many of you know, with one of these modalities as an exemplar for how we hope that progress in this field can be accelerated. We've been working with antisense oligonucleotides now for the last 5.5, 6 years. And by way of scientific background, again, covering material that's well familiar to many of you, these are short snippets of synthetic DNA or RNA that have been worked on for some 3 decades. And over those 3 decades, multitudes of folks, including many critical figures like shown in the bottom right here, have demonstrated that these can be quite effective drugs, first in cell models, then in animal models, and now increasingly in patients. Generations of chemistry have been applied to modify these synthetic DNAs and RNAs to give them good drug properties, such that they are now currently simple to manufacture, relatively easy to deliver, at least in to certain organs. And these ASOs, for short, can be used to bind to different genetic targets, to alter patterns of gene splicing, or target certain RNAs for destruction. And in many cases, those are very, very useful tricks for designing a therapeutic strategy for your disease. The exemplar case that many in the field have been following now for many years is the use of an ASO for spinal muscular atrophy. This was demonstrated through really heroic work beginning with Yimin Hua, Adrian Craner, extending out to Frank Rigo, Frank Bennett, and many folks at Ionis, and many talented clinicians who showed that an 18 nucleotide synthetic RNA, an ASO, dissolved in artificial CSF, could be given to infants with spinal muscular atrophy by spinal tap 3 times per year. And it could be used to take advantage of the fact that patients with spinal muscular atrophy have a backup gene called SMN2 that can be targeted with this type of drug, and that backup gene could be refurbished and induced to work again. This is an evolutionarily inactivated copy of a gene which is luckily present in all of us, but using an ASO, one could change the splicing pattern of this gene and render this previously inactive gene active again. And this clinical trick has had wonderful benefits for patients who are with spinal muscular atrophy. This early effort has really spawned a wide host of many additional promising ASO drugs that are some approved and many in trial now underway. And among those that are FDA approved are ASOs for spinal muscular atrophy, TTR amyloidosis, familial hyperchylomicronemia syndrome, and there are many other ASOs that are in various stages of trial for ALS, Huntington disease, Angelman syndrome, Dravet syndrome, inherited retinal disorders, and more. Now, many of us are here because of an interest in mutation-specific genetic therapies. And to highlight what's different about these sorts of therapeutic approaches, I've mentioned that Spinraza or nusinersen, the drug for SMA, targets an evolutionarily acquired mutation that's found in all patients with SMA. But unfortunately, this situation is actually the exception, not the rule. What happens if the target mutation is only found in some individuals with the disease, or perhaps even only in a single patient or a very few number of patients? How do you test it? How do you develop it? How do you tell if it works? And what is the proper way to regulate this type of activity? These are questions that used to be theoretical, but we had the opportunity to actually put some of these questions to the test. Back in 2017, many of you know, we met a young girl named Mila, and through a series of fortuitous events, worked hard with a collaborative team to develop an individualized ASO for her specific mutation, for her specific ultra-rare genetic disease. And she had been diagnosed with something called Batten disease, an ultra-rare condition that impairs the ability of the body to clear cellular waste. This seemingly subtle impairment leads tragically to progressive neuronal cell death in the brain, and it leads to children developing progressive vision loss, ataxia or incoordination, cognitive regression, and is ultimately fatal by around age 11 in children who manifest this condition. And as this unusual case proceeded, we became connected to this family and discovered that she had an unusual genetic mutation. Her normal CLN7 gene is shown on the top. Her CLN7 gene is shown on the bottom. A whole genome sequencing that we performed showed that she had an unusual mutation, a misplaced segment of genetic code in the middle of this gene that created a new splice site. And this unusual mutation created for us an opportunity to design an antisense oligonucleotide expressly to correct it. This was a strategy that hadn't really been contemplated before, the idea of doing this type of mutation-specific therapy for a specific individual. And it was necessary because at the time there was no available, say, simple gene replacement therapy for CLN7 Batten disease. Thankfully, in the years since, such a gene replacement strategy had been developed. But as we know, those types of efforts take a long time. So at the time when we met our patient in 2017, this was the only option available, and we pursued it. We designed an ASO to correct this. using computational algorithms to target our sequence to the area of Mila's genome where missplicing was occurring. We performed a drug screen using our patient's cells, and were able to find drugs that were able to restore splicing patterns from the mutant splicing pattern back towards the normal splicing pattern. And we were able to show that application of this drug to her cells, at least in a dish, made them significantly healthier. Shown in red were the abnormal cellular wastes that were accumulating in the cytoplasm of her cells in the absence of treatment. And shown in green are what happened when we treated her cells with this ASO drug. Just at 24 to 48 hours of treatment made these unusual white bubbles that are normally representing the accumulation of wastes. We saw those bubbles disappear, and really convincing us that this treatment showed strong, plausible proof of concept. And with evidence like this, we were able to work collaboratively with the FDA to orchestrate accelerated manufacturing and safety tests and complete those in a matter of short 3 months, and then administer this drug with some clinical impact and improvement on quality of life for almost 3 years. This effort really has opened up a new realm of possibilities for orphan conditions, for conditions even as rare as one. And it's led to a draft FDA guidances released now amazingly 2 years ago from the FDA, giving guidelines for how the FDA is willing to accept submissions of antisense oligonucleotide drug products for these types of severely debilitating or life-threatening conditions, even when there's only as few as a single patient. And it has paved the path towards many other case examples that are gradually working their way through the system. We ourselves have talked publicly about a young infant who was accidentally diagnosed with a condition called ataxia telangiectasia at birth. She actually had an abnormal newborn screen that indicated a concern for a potential immunodeficiency, but it turned out that that was a false positive. She did not have immunodeficiency. Instead, she had, arguably, however, a worse problem, a neurodegenerative condition that leads to progressive loss of coordination and leads to children being wheelchair requiring by age 10, and unfortunately unable to control their eye movements, their swallowing, and protect their airways. It's a very tragic condition that is fatal by young adulthood. And while there were no available treatments for this young infant that were in the pipeline, we recognized an opportunity to intervene because one of her mutations created a new splice site, just like the mutation in our Batten patient had created a new splice site. And we were able to use the same approach to design an ASO to block this missplicing, and then to advance an experimental ASO for her and begin treating her by age 2. This trial, which we are actually just beginning to report, we have a manuscript that in submission describing this, has given us 3 years of optimism to date. Today at age 5, our patient's doing quite well. She's doing quite stable on clinical rating scales. Her brain volumes have been approximately stable. There's often significant degeneration in this condition. And while her cerebellum is slightly smaller than normal, it does appear to be on the mild end of this condition. And various biomarkers also give promise that this early intervention might be working. I'll also note that this individual patient trial began as an individual patient trial, now has the prospect to expand. We've identified a second patient with the same mutation, the same condition, and actually, coincidentally, the same age. This patient was in Germany. We started treatment in Boston, but now we've gotten permission to return her back home to Europe, where she continues in the experimental trial at a distance. And we've got our sights set on several additional patients with the same mutation in various parts of the world that we are also trying to expand our experimental trial to. Here's just another case example of a third case. This is a 5-year-old with vision loss and pain insensitivity. She has a genetic condition that has a name that only neurologists would love. It's called posterior column ataxia with retinitis pigmentosa. Even I have trouble saying it. The key features of this condition are that it causes vision problems. That's the retinitis pigmentosa. It causes problems with sensation. That's the sensory neuropathy. And it causes ataxia or incoordination. And it's caused by mutations in a gene called FLVCR1. And the critical factor is that the children with this condition, they have sensory problems that make it difficult for them to feel pain. And in fact, it actually leads to a lot of inadvertent self-injury because they don't realize that they're hurting themselves at a young age. Eventually, however, they learn to manage that and they learn how to protect themselves. However, then the later phase of the condition is really the main concern is the progressive vision loss that they experience. Our patient here has had progressive restriction of her ability to see. She's losing her peripheral vision, and her central vision is getting fuzzy. And so we have been working to try to develop a treatment for her. It turns out that her traditional clinical genetic workup was inconclusive, but whole genome sequencing revealed that she had mutations in FLVCR1 that included one very complicated mutation, the second one, insertion of a 3 kilobase mobile DNA element, that had, in essence, the same missplicing consequence as I've already described for the previous 2 patients. And Bo Zhao in my laboratory was able to demonstrate that this could also be corrected with a splice-blocking antisense oligonucleotide, a patient-customized ASO. And in discussion with the family, with her clinician, and with regulatory authorities, we considered the fact that with her progressive loss of vision, the fact that ASO dosing to the eye is actually relatively well established. We posed the questions of whether or not treatment could slow progression of her vision loss and improve her quality of life with reasonable clinical risks, and obtained agreement from all parties, importantly, first and foremost, her family, as well as her clinicians, Dr. Austin Larson and Emily McCourt, and the FDA. And working in concert with the NLORM Foundation, we've completed manufacturing and safety studies and hope to be filing an IND to submit this trial for consideration to the FDA very, very soon. So these are just a couple of case examples that I've highlighted in these slides, but there are several others, that, some of which are published already. From our initial report of intervention for a patient with Batten disease in 2019, there have also followed similar efforts, including one from Columbia and Neil Schneider's group with an antisense oligonucleotide for a patient with ALS. And an additional remarkable effort from Bob Brown and Jonathan Watts at the RNA Therapeutics Institute, where they also designed an antisense oligonucleotide for a different genetic form of ALS that has been showing very, very promising results. And this type of effort, it's been very gratifying to see, is just indicative of just the amount of activity in this space. And folks in this audience know very well, and at AgCells know very well, how much interest there is in this space to develop these types of orphan therapeutics. I'd like to acknowledge that the work that we started with Milasen has really been taken up by many parties. This began, I would say, first with the Oligotherapeutic Society, which assembled a rare disease task force in April of 2020. And put together a first scientific briefing document representing scientific best practices for this early type of work. And growing from the Oligotherapeutic Society, there's now a pretty remarkable ecosystem, which has materialized now in the last 5 years. And just some of those parties are shown here, from the FDA, which continues to engage in this with frequent conversations, draft guidance releases. They've really taken to this area and have been very supportive of this area. Oligotherapeutic Society. I've also mentioned the NLORM Foundation, a nonprofit started by IONUS. Many academic institutions, including governmental, NINDS and NCATS, our hospital, UMass Medical School, University of Leiden and the Dutch Center for RNA Therapeutics, various ethics groups, including the Center for Bioethics at Harvard Medical School and NYU. And then gratifyingly as well, we are seeing companies also stepping up to help patients in this space, including IXLs, as well as companies like Crayon Bio, Perlara, La Jolla Labs, and Everyone Medicine. We ourselves have formed coalitions of academics and investigators like the N of One Collaborative in the United States and the One Mutation, One Medicine group in Europe, and have been gratified to have received the support of foundations like the Chan Zuckerberg Initiative to help catalyze conversations here. I'd like to close by just one cautionary comment that as we've seen this space really explode, it's fair to say that caution is still required. And I'm trying to advance, but for some reason, there we go. That this is a space for which hazards still remain to be navigated. There are still many unknown unknowns. And so we have to be careful to keep in mind that, that there's still much to be learned about these technologies, and that when we choose to deploy them and to advance any one of these drugs into trials for any of the children that we care for, that we're always carefully weighing the disease risks against these potential and sometimes as yet unknown hazards of this type of treatment chemistry. And by way of example, a particular study that has drawn some attention in the media that we began discussing last year and was covered in the New York Times recently is an important and sobering reminder that I'd like to highlight. And here's an example of an ASO that we developed for an infant with life-threatening epilepsy. This is a child with KCNT1 epilepsy, one of the worst epilepsies that we know. Patients with this condition have very, very early mortality. Many die as young as age 3 because they suffer dozens of seizures per day. This particular condition, early infantile migrating focal seizures of epilepsy, is caused by a mutation in a gene called KCNT1, which creates a poisonous channel that allows in much too much current and makes neurons fire uncontrollably. We had developed an antisense oligonucleotide to help quiet this gene and dosed a patient with this experimental drug on day 0, on day 30, on day 40, and on day 0, on day 28, day 48, and then shortly after on day 56. And we saw a remarkable suppression of her seizures down from 5 to 20 per day, down to 0 for almost 50 days solid. After this 4th dose, her seizures went to 0. They stayed low, and then they gradually returned as the doses were held. And the reason that the doses were held was that at the same time that her seizures improved, we also unfortunately encountered a serious side effect in this patient, where she developed hydrocephalus, a condition also known as water on the brain, an expansion of the water fluid-containing compartments of the brain that can be quite serious, in some cases life-threatening. And that this was serious enough to actually require neurosurgical intervention for this patient. It caused us to pause the trial and talk to folks in the field to inquire whether this type of side effect had been seen before. And while it had not been in a previously established definitive side effect of antisense oligonucleotides, our conversations with many folks revealed that dose-dependent hydrocephalus had also been seen in a Huntington's trial conducted by Roche with an ASO made by IONUS. And this revealed to us that there are still these as yet to be determined unknowns that we may still encounter. We believe that this is a new and important potential toxicity of some high-dose ASOs that we have to watch out for. Now, luckily, we do believe that this is something which is monitorable and intervenable. For instance, in this case, where this patient had a VP shunt placed, which was able to reverse the hydrocephalus. And in this particular case, weighing the risks versus the prognosis, the FDA and our IRB have approved dose reduction and restarting the trial with careful clinical monitoring. But it is just a very important and healthy reminder that as we push forward, that we have to keep our eyes and our ears open and collaborate to try to surface these types of potential hazards so we can avoid them as quickly as possible. So thank you so much. Let me stop there. And I want to acknowledge the many folks who have been involved in this work over the last 5 years, especially the support of many patient and family foundations in the rare disease space, shown here in the right column, as well as generous support of the CZI, groups like the Harrington Discovery Institute and the NIH and the Human Genome Research Institute. Thank you very much.